All right, so today we're going to talk about the origins of life. So we're going to go through a few eons, then we're going to break it down just a little bit more. But like I said, a lot of what you're going to see today, we have already seen, probably in a little bit more of a condensed version. So up here you have the Hadean eon, and this is when Earth forms. There's no oxygen in the atmosphere, and there's constant volcanic activity. So when there's no oxygen, what term do we use? We talked about this already. No oxygen. Begins with an A.
Abiotic.
AN. We talked about it when we did photosynthesis and respiration.
Anaerobic.
Anaerobic. There you go. Good job. Then we get to the Archean eon, and this is when we had our actual first anaerobic prokaryotes. Then came along the first photosynthetic prokaryotes, and then the oxygenation of the atmosphere, which essentially caused mass extinction of the anaerobic bacteria. Next is the Proterozoic eon, and this one is when we saw the first eukaryotes. So now we're seeing a difference. Okay, so we had prokaryotes prior, and now we're seeing the eukaryotes pop up. We're going to talk about this a little bit later in the presentation, but what's the difference between a prokaryote and a eukaryote? Which one has one?
The eukaryote.
The eukaryote has a nucleus and the prokaryote does not. Very good. We're going to see some other differences that we've also talked about as we go through the presentation. This is when the first sexually reproducing organisms came about, also the first multicellular organisms, and then a glaciation event. So this is where we see glaciers pop up. Then we have the Phanerozoic eon, and this is when we had a major diversification of life. So now we see a diversity of life. Major mass extinctions eliminate 90% of marine species, and now we have the modern era. So now we're going to get into some pieces that we've already seen and talked about, whether it was within evolution or earlier in the semester. But the fossil record provides evidence about the history of life on Earth, and we saw this the other day. So where do we find our oldest fossils? The farthest down, okay? And then over time, okay, they keep building, and we'll find our youngest ones at the top. We also know that fossils are remnants of something that was once living. And typically when we hear the word fossil, what's the first thing that pops into mind? Dinosaurs, okay? So with studying fossils, we are able to see how organisms have changed over time, because this was one of our big pieces of evidence for evolution. The next piece to this is that paleontologists are scientists who study these fossils. So people do this for a living. And when they're studying fossils, they move to another piece of evidence that we looked at with evolution, and they start to look for anatomical similarities and differences. And we talked about 3 different types of structures that show evidence of common ancestry. So who remembers those?
Vestigial.
Yep, so what are the other 2 though? So vestigial and then, yep,
Homologous.
homologous, very good, okay? So homologous is what?
Same structure.
Yep. Different function. Analogous is?
Different structure, similar function.
Yes. And then vestigial is?
Vestigial is something that doesn't do anything.
Perfect, okay? Next, you're gonna just see, this just shows us that anatomical deals with the anatomy. So when we're looking at anatomy, we are looking at body structures. Another piece here, and we're actually gonna get into this tomorrow once we start classification. So up here is a cladogram or a phylogenetic tree. And this is also what shows us how closely we are related to other organisms. So up here shows us different types of plants and how they are related and how they have evolved over time. Up here, and this is what we're going to delve into a little bit with the endosymbiotic theory, we have a prokaryote and a eukaryote, and y'all already told me the major difference is nucleus versus no nucleus. So the prokaryotes don't have a nucleus and the eukaryotes do. What's the other big difference that we had talked about a while ago? Perfect. Eukaryotes have membrane-bound organelles, and that's going to come into play today. So going back to fossil evidence, there are 2 ways that we determine the age of a fossil, which is super, super important. The first is relative dating. And you'll talk about different things like this more in a physical science class and sometimes even in chemistry. But relative dating is what allows paleontologists to estimate a fossil's age. And they do this by comparing them with other fossils. And obviously we know, and we've already said today, that the oldest fossils are on the bottom and the youngest ones are at the top. The second piece to this is absolute dating. And this deals with radiometric dating. So technically what scientists are studying are radioactive isotopes. And that's why I say this is things that you would study in physics or chemistry. And this is a big piece that they even get into at the college level. So they're able to determine the exact age of a fossil by doing these things. So we're not going to get crazy into depth with this, but this is a whole mathematic equation where you have to calculate half-lives and all kinds of stuff with radioactive isotopes. So taking us back to some other terminology that we already kind of reviewed a little bit earlier is anaerobic versus aerobic. So anaerobic, we said, is without oxygen, and aerobic is with oxygen. Also prokaryotes versus eukaryotes. So we went over 2 important things, the nucleus and membrane-bound organelles versus no nucleus and no membrane-bound organelles. Also, we know that eukaryotes are much larger, okay, and they are also very, very complex, whereas a prokaryote is much smaller and much more simple. Okay, so this is comparing our bacterial cell to a plant or animal cell. So now we're going to talk some timelines. So we talked eons earlier, now we're going to jump into some timelines. But this is pretty much what we've already seen earlier today. So the Earth formed about 4.6 billion years ago. This is when there was an anaerobic atmosphere, meaning no oxygen, and it had simple organic compounds, okay? But super important, there was water that was present. So how did we continue to go on with this, and how did life actually develop on Earth? So there is what is known as the Miller-Urey experiment. And this showed that the mixture of complex organic compounds was necessary for life. But it could form from simpler compounds. So up here, you see some examples of some complex compounds. So you see things like ammonia, which is NH3, CH4, which is methane, H2O water, and H2, which is hydrogen. So now we're going to get into some important events about life and how it developed on Earth. So up here, about 3.5 billion years ago, we saw the first anaerobic organism, okay? So the first anaerobic organism, they were prokaryotes. So everybody here, which came first, prokaryotes or eukaryotes? Prokaryotes, okay? And these were able to thrive in the harsh conditions of our early Earth, because obviously we didn't have everything that we have now, and times were definitely a lot different. So they were able to survive in these environments. Then as time goes on, so now we're 3.4 billion years ago, now we have the anaerobic photosynthetic organisms that start to evolve. So we're going piece by piece. So what's the purpose of photosynthesis? What does it produce? It produces oxygen. So now we said we were an anaerobic Earth, right? So after the evolution of that photosynthetic bacteria, now we are going to have oxygen. So now the environment and the atmosphere is aerobic. So previously it was anaerobic, but now with these photosynthetic organisms, we are able to be aerobic because now we have more oxygen. So now we have aerobic prokaryotes. So we're still hanging with the prokaryotes. So first we had anaerobic, now we have aerobic. So about 2 billion years ago, we get aerobic eukaryotic cells. So this is where I had mentioned the endosymbiotic theory comes into play, and you're going to learn a little bit more about this in your Edpuzzle today. So this explains how the first eukaryotic cells developed. And this deals a lot with membrane-bound organelles, specifically the mitochondria and the chloroplast. So with the endosymbiotic theory, there's a symbiotic relationship that's happening among different prokaryotic organisms. So now, what we know as prokaryotes became permanent organelles, which you see happening in the diagram at the bottom over time. So these are our evidence of symbiosis, endosymbiosis. So the mitochondria and the chloroplast, like I mentioned on the previous slide. So they contain their own DNA. And these are both membrane-bound organelles. The second piece of evidence is that the ribosomes of these organelles are similar to those that we find in bacteria. So remember way back when we did prokaryotic and eukaryotic cells, we talked about some things that we found in both. And then we talked about things that are specifically to prokaryotes and things that are specific to eukaryotes. So ribosomes is one of those things that we would find in both. Next, we have our mitochondria and chloroplasts that reproduce by binary fission. So this is just like bacteria. How do bacteria reproduce? Yes, which is known as what? What type of reproduction? Asexual. There you go. Perfect. Okay. So asexual reproduction. So binary fission works just like that. Okay. It splits into two new organisms. So both bacteria and the mitochondria and chloroplasts have DNA that is circular. And then both bacteria and mitochondria and chloroplasts reproduce by what is known as binary fission. So asexual reproduction. And then one of the last pieces, sexual reproduction develops. So this is some other pieces that we've already learned. So the crossing over and shuffling of genes that occur, which is a way that we get what? Crossing over and shuffling of genes. How we get what?
Genetic variation.
Genetic variation. Perfect. Okay. So with crossing over and the shuffling of genes, this allowed for diversity to happen. So once this happens, now we have multicellularity. So multicellular organisms obviously have more than one cell, and we're used to talking about bacteria, which are single-celled organisms. So now cells have specific jobs. So we have all these cells in our body that now have all these specific jobs. So the same thing has happened within other organisms as well. So this is what leads us to what we currently see in our own bodies. So muscle cells, liver cells, brain cells, kidney cells, bone cells, nerve cells, and the list goes on. So now we're just going to recap briefly what we saw today. So we're going to take these 5 events and we're going to put them on the timeline. So which one do you think is going to come first? Anaerobic, very good, organisms. So anaerobic organisms are going to come first. Okay, what's going to come next? Which one comes next? Which one? Photosynthetic anaerobes, because we're still sticking with anaerobic, right? Because then photosynthetic anaerobes gave us what? Oxygen. So now we have what?
Aerobic.
Aerobic prokaryotes, very good. And then next on the timeline would come eukaryotes, very good. And last would be multicellular organisms.